Hello, everyone, and welcome to the third part in our webinar series sponsored by Biogen. This taking us from discovery through to the clinic, and we're in the middle of it right now with the one that I'm sorry that we've titled it, Oh, the Humanity, but really trying to take a look at human ALS lab models. And we have an unbelievable panel with us today. So I will stop sharing my screen so that you can see everyone nice and large. Hello, we have on the line with us right now, Dr. Rita Sattler and Dr. Clive Svenson. Our other two panelists, I think, are in the active process of coming on, but I would like to first start by introducing the panelists. We're going to start with Dr. Rita Sattler, who is an associate professor of neurobiology at the Barrow Neurological Institute. Many of you will also know that she was assistant professor at Hopkins before, and you will know her from her work in neurotransmission in ALS, synaptic pathology, as well as RNA editing. Highly encourage you to go see the more recent ADAR2 story that's been published on her work with that. So welcome, Rita. Thank you so much for being here.
Welcome, Dave. Thanks for having me.
Yeah, so because these things we do ask afterwards if people are okay to let it stream. We want to, even though this is aimed at a scientific audience, we have a scientific audience out there. Also love to ask people what kind of got them towards ALS. Like I know you worked with Michael Tymiansky in Toronto, and then when you moved to Hopkins, was it the excitotoxicity piece that like Jeff was working on, Jeff Rothstein and others, that sort of pushed you towards ALS, or was it something else?
No, it was actually, I started at Hopkins with Dr. Rick Huguenier because I was very interested in learning more about the regulation of glutamate receptors, which is what I studied when I was in Toronto and looked at mechanisms of stroke and the role of glutamate in excitotoxicity in stroke. So I joined Rick's lab in order to really dive deeper into the regulation and the molecular mechanisms of glutamate receptor regulation, and really still trying to understand the glutamate homeostasis and its role in both health and disease. And then from there, I sort of became more interested in whether this excitotoxicity is relevant for other neurodegenerative diseases as well, including ALS. I actually wrote my very first ALS grant when I was a postdoc with Rick in Rick's lab, and that's how I sort of learned a little bit more about what was happening across the street in Jeff Rothstein's laboratory, and then really started working with Jeff when he started a small startup company that was trying to identify small molecule compounds to regulate this glutamate homeostasis in order to show protection for ALS. So that's how Jeff and I actually got to meet each other, and that's how I really got introduced to ALS.
And then we grabbed you and we
pulled in. I never left.
That's what happens with ALS, yeah. Great, well, thank you so much for being here. We also have on the call, we were gonna go alphabetically, so we'll skip back to Dr. Victor Rayfuse, who is can we hear you, Victor?
You hear me now?
Yes, we hear you great, thank you. A professor of medical neuroscience at Dalhousie University. Another individual on the call who in Canada we really go to in terms of knowing the scope of where we've come from in terms of motor neuron development from embryonic stem cells, iPS cells, and really where we are in the current state at any given time. So thank you for being here, Dr. Rayfuse. I also urge you, recent paper on terminal Schwann cells and ALS models and different muscle types. So please, audience, go out there and have a look at his recent paper. Victor, I just have a quick question. So you started with Tessa Gordon and then moved into Lynn Lampmaster's lab working on neuromuscular junction. Was ALS sort of on your radar before then, or was it not until you got to Dalhousie and started working on the motor neurons with the embryonic stem cells?
Yeah, no, coming out of Lynn's lab, if anybody knows Lynn Lampmaster's work, she's basically a neurodevelopment person, particularly the motor system. And so I started working with ES cell-derived motor neurons as a model of neurodevelopment. I wasn't even thinking about any diseases associated with it. And then ALS Canada actually invited me to give a talk. And then it was like, I was dumbfounded of, how naive I was. So, I immediately started to gear a lot of my research towards ALS. And I still do neurodevelopment. I'm still a neuromuscular physiologist, but really that was my first introduction to the field of ALS, and continue to examine it to this day. Yeah.
Thank you. Great. Well, thank you for being here. And last but not least, on the group that we have on the call, we have another stem cell pioneer, Dr. Clive Svenson. I urge people out there to actually go back to 1995, where you have a Neurons from Stem Cells paper, which is a great and fascinating read. Very before things really got ramped up, thinking about this, and he is a professor of medicine and biomedical science at Cedars-Sinai, also a Sheila Essey Award winner, the Carey and Simone Vicker Family Chair in Regenerative Medicine. And also, many of you may know that beyond the work that he does with advancing where these types of cells, a most recent review paper I urge you to read as well on multi-lineage cells and all the different technology, head spinning technology, body on a chip, bioinks, all these things. Go have a look at that. Also, currently has evolved all the way from bench to bedside the concept of BDNF and the amazing astrocyte, and being able to use the technique from Nick Boulos and alongside Bob Balow to intraspinally inject astrocyte-driven cells that are expressing GDNF in patients with ALS. So wonderful to have you. Thank you so much for being here, Clive. In terms of ALS, I know you have a very broad set of foci, but have been really working on ALS as well for many years. Was it in Cambridge that you first started to think about ALS, or was it when you got to Wisconsin? What sort of connected you to this particular field?
Actually, no. In Cambridge, I was much more involved with Parkinson's disease, and I think that review was with Austin Smith, who's my buddy in England. Very, very long time ago in TINS. But yeah, we were thinking about stem cells. The issue back then was actually for Parkinson's was fetal tissue transplants. And I was working with Steve Dunnett and Anders Bjorklund and Uli Lindvall as a postdoc. And they were trying to get fetal tissue. And the big issue was they needed 5 fetuses for one patient. And the technicalities of that and the ethics were amazing. So we decided to try and get stem cells from the area of the brain that makes dopamine neurons. And we thought it'd be easy. And we just thought, oh, we'll take the cells and we'll expand them, and now we don't need fetal tissue. And that really introduced me to the complexities of growing cells in the petri dish is very different from in the developing brain. So then wind forward, I came to Wisconsin. I can still do the Wisconsin accent, and I'm a Badger fan for 10 years in Wisconsin. And I was introduced to someone there through Lucy Bruin, who was at the ALS Association, called Jeff Kaufman. And Jeff Kaufman, I met him at his house in the late stages of ALS, and it was around when Bush was banning embryonic stem cells. And he just pointed he couldn't talk, he just pointed his pointer, and he had a word for the politics then and the people in it, and it was dumbass, because they wouldn't allow, the Bush administration wouldn't allow embryonic stem cells to be used to treat diseases like ALS. So he was very frustrated. I met Jeff, and that was my introduction. And so through the ALS Association, I got involved, and I've been involved ever since. Like Rita, you see this disease, and we're all the same. You see a patient. Yep. It's a nice person disease. It happens to the nicest people, and we don't know why. And that just drove me for the last 10 years to understand more.
Sure.
I think Lucy deserves some credit for pulling a lot of people closer to the field over the years. And I think I noticed you said you were a Packers fan. That me.
I'm sorry, you reverberated, David.
Oh, sorry. I think you said you were a Packers fan for 10 years. Is that going to anger anybody? Because it kind of means it stopped at some point.
I was British. It was so bloody cold. I couldn't understand how so many people could, but I loved it. No, I was. My son is still a Packers fan.
Okay. Well, there you go. You passed it on. So I would love to. So I'm going to just get right into it. Be mindful of your time. When I thought about doing these, when we were thinking about putting together this panel series, I really wanted one on this, and there's kind of the reason why is because, there's still, even though there have been huge advancements in terms of where we're at in terms of modeling human cells in the laboratory, it seems like ALS is still at a point where we haven't fully embraced it. There's some people who say you need to have them for your studies to prove that this is in a human cell. There are others who really embrace it. And then there are others who would rather do something in a HEK cell or an NSC cell, or they really value Drosophila or yeast. But, you know, when it comes to these ones, they start to get a little bit uncertain. Maybe it's because of the fact that they don't always represent, at least an iPS cell doesn't always represent an aged cell type, which is obviously something you've all had to discuss a million times over. But, you know, and I'll digress, and we'll introduce Dr. Egan after. But just starting out on that, where are we today versus where were we in 2020 in terms of these types of cells, going from ES cells to iPS cells to eye motor neurons, and glial cells even, but we'll get into that a little bit later. And then where does this fit into the ALS spectrum in terms of, should things be done in rodents, in your opinion, be validated in these types of cells? Is discovering these types of cells, should they be going straight into clinical development? It's a loaded question, but you know, just kind of getting a state of the nation of where we are in terms of these cells. Who would like to start? If not, I'll pick on somebody. I won't pick on Kevin because he just showed up and he probably didn't hear the whole question.
I mean, I can maybe start the discussion a little bit about this, because what I often do when I either speak to my own students or when I talk to other students about using iPSC as a model system for not just ALS but other neurodegenerative diseases, that I encourage them that if that's what they would like to do, to always really think about how can they use other models in addition to the iPSCs, because other models have pros and cons too. We know that the rodent models have, you know, a lot of advantages, and we learned a lot about the disease, not just, again, ALS, but other degenerative diseases as well. But they also have a lot of disadvantages, and we know that the translational aspects from the animal to the humans have really not been very encouraging over the last years, which is one of the reasons that probably some of us decided, well, if we have something that's more a human-like model, then, you know, we can put something in between the rodent and the patient before we even go into the clinic. But there's other models too now. We have very strong and good fly models. We have zebrafish models. We have yeast models even that are being used to study mechanisms of the disease. And so what I usually tell people that, you know, if you have just one model, you're probably not going to be good off with that, because you might, you know, be chasing an artifact of that particular model that you're looking at. So having an opportunity to introduce maybe 2 or 3 models, if you're really trying to validate the findings that you have, I think that's the best way to go about it. And then you can overcome the difficulties that we have with the iPSC, such as the fact that we don't really work in an aged environment, right? We know that whatever cells we're differentiating, they're very embryonic-like, and I don't think anybody's going to dispute this. And so, you know, in that regard, you know, it's hard to maybe look at aged aspects. Nevertheless, we do see phenotypes in these cells that are very significant and that are very relevant, and that we can validate either with other models or post-mortem tissue samples when we look at this. So I think there's not an easy yes or no whether this is good or bad, and I think there's also, people have different opinions on this, and but I think as long as you are aware of the fact that there are disadvantages, then and you can control some of these, and you can, you know, sort of overcome these disadvantages, then you know, it's a very good model to use. It's expensive, so you're going to have to be aware of the fact that, you know, it's also, people like to go to 293 cells because they're cheap and they're fast. So I don't know, Clive.
Yeah, I agree. Just about everything Rita said, and you know, I think they're all models, right? The thing that we're focusing on in my lab, and generally I encourage my students, is validation. So if you can validate a yeast model or a fly model in a human, and I think Kevin got the closest to that with his C9 data that he did with the drug company. So you find a hit and you try and validate it in patients. That's where the rubber will hit the road. And I don't care if it's a fly model, a yeast model, an IPS model. If you validate something, that's going to be the key. And I think Kevin's model, it was a complicated process. I know, Kevin, in your trial, but that was the right approach. And if we can validate, then I think drug companies will start using those models that get validated. Right now, it's a horse race. And, you could put money on IPS or you could put money on yeast, but we got to find out what's validated in humans. That's my feeling.
Okay, so, oh, sorry, Vic, yes, please.
No, before you introduce Kevin, I'd just like to say, sort of looking in from outside, sometimes the field and being a participant in the field, I think there's been a lot of changes in the field over the last 10, 15 years. And I think there's a lot, originally, I think they were used more as just a cell survival, cell death assay. And I think we've learned a lot more. You can do a lot of electrophysiology and look at the effects of drugs on how it changes membrane properties, firing properties, and things like that. The introduction of all sorts of other cell types and how that affects motor neuron survival and behavior. So I think, in some sense, we're still learning a lot about what these cells can do. So, but I agree with everything that's been said. I mean, validating in various models is a necessity. I firmly believe that.
Great. So actually, I'll take this moment to introduce our fourth panelist, Dr. Kevin Eggen. Thank you so much for being here. He's a Group Vice President, Head of Research and Early Development of BioMarin, but also a professor in the Department of Stem Cell and Regulatory Biology at the Harvard Stem Cell Institute. A MacArthur Fellow, Director of Stem Cell Biology at the Stanley Center for Psychiatric Research at Broad, and also several landmark things, including not just around iPS cells and embryonic stem cells, but recently many of you out there will know about being a co-discoverer of Stathmin-2 as a very interesting treatment target from his lab, and also the recent work that's evolved over years with the C9orf72 knockout models in the splenomegaly, resulting ultimately in looking at gut-brain axis involvement in ALS and environmental impact. So thank you for being here, Kevin, very much. We had asked the others what led them into ALS research. I know you have worked with a lot of heavy hitters over the years, but was it once you got to Harvard that you sort of got pulled a bit into this, or was it just the opportunity that this was a good disease to work on deriving iPS cells into a cell type from patients, or what led you to ALS?
Yeah, and hopefully I'm coming through okay. I'm sorry that I've been battling connectivity issues all day from my home. I'm coming to you from the historic quarter of downtown Boston, and sometimes I feel that the internet cable connectivity is from the revolutionary times. So I apologize for any discontinuity that I might have. Thanks very much for inviting me to be here. I'm sorry about the delayed joining. My story for getting into this really starts with Dolly the sheep. And when I was thinking about where to go to graduate school in the late 90s, was just when Ian Woolman and his colleagues were really reporting on Dolly. And I became really fascinated in understanding how that worked, and went to Rudolf Janish's lab at MIT for my PhD, predominantly to work on mechanisms of reprogramming and really trying to understand fundamental mechanisms that governed the state of cells, why they have the biologies that they do, and how immutable those phenotypes are or not. And really during that time, of course, Jamie Thompson at University of Wisconsin reported the first human embryonic stem cells, almost within months of Dolly. And so everyone became quite excited about the concept that we might be able to make sort of replacement cell therapies that would be genetically tailored. And while lots of my colleagues really became drawn to that, I've been, since I was quite young, interested in genetics and genetic disorders. And I became really fascinated by this kind of concept that maybe we could make cell types that just had never been accessible to us before. And, I had Bob Weinberg on my thesis committee, and, I think seeing what they could do in oncology with the disease being taken out of a patient and given to you in the lab, I think was really striking for me. And of course, in nervous system disorders, the stuff that you want to understand and study is gone by the time that patients are really coming to you. And so the idea that you'd be able to make human material, to be able to do experiments like folks have in oncology, I think is very attractive. And so that really took me to the Harvard Stem Cell Institute, close collaborations with many people there, including human embryonic stem cell derivation, at first closely with Doug Melton, and then work on reprogramming, which, I always wanted to take in this area. And the question was really in what direction. And, at the time, really, I would say the only type of cell that we really knew how to make really from stem cells were spinal motor neurons. And I think early entry into the field meant I really only had one choice of what I could reasonably work on. And, and I really credit Tom Jessel, Henrik Wichterle, and Chris Henderson, who was director of the Motor Neuron Center at the time, as being key to kind of my recruitment to the field. That plus, I would say, encouragement from all the other panelists that are here. But what really kept me in the field was Bob Brown, and Bob really taking time to introduce me to patients, and I think to really integrate me into the patient community and familiarize me deeply with the importance of this problem. And it's really, I would say, those two things that that put me in this chair today.
I think you're muted.
What a host. I think it's quite interesting to note that all of you were impacted by, meeting people with ALS and being connected in through that way to really want to double down on working in this field. It's a testament to the not only the work itself that can be done in the field, but also the power of the disease itself. So Kevin, so while we're on there, the discussion had been around, there's still a lot of naysayers in terms of the value of iPSLs for whatever hurdles they have to overcome. But what are some of those hurdles that they have overcome in the past decade and a half or decade and a little bit that in 2020 makes them, much stronger than they were, say, in 2008?
Yeah, that's a great thing. Stem cell technologies have gone through the classic kind of, innovation phenomenon where there's the burst of expectation, the trough of despair, and then the, kind of rich period of delivery from a new technology where, everyone was really excited about the promise, with Shinya Yamanaka and colleagues' discovery of iPSL reprogramming. Fortunately, folks like Clive and I and Henrik and others, had been working on some of the cell modeling already, so that we kind of were somewhat ready to receive this. But I think the challenges early on were almost entirely chalked up to the fundamental power of the system itself, and that early on, while it was amazing that these stem cells had enormous replicative capacity and that they could make all sorts of different cells, that was also the challenge of them. And the cookery book that we had of methods for really making particular cell types and then isolating them for study was not very good. That is to say, you could do it, but it had not yet been refined enough that you could do experiments with it. And so a lot of that, kind of trough of despair, I think was really, given over to the challenge of run-to-run reproducibility of differentiation schemes, a real understanding that, most of the motor neuron differentiation schemes early on were making a bunch of different cell types that you would expect to find in the spinal cord. But, I think not an ability to really make just one type, which is what people's expectation was. And so there was a lot of variation in early experiments that wasn't so easy to understand. And then on top of that, most experimental biologists aren't used to dealing with the complexities of human genetics. And it can be easy to think about, a human genotype as something that's kind of monolithic, but, nobody knows better than Rita that that's not true. And that, we're used to sort of thinking, well, if we make this mutation on black 6, it'll kind of be the same. Even that's not really true. But as you start making studies of a variant in a human genetic background, you've got to contend with all the other genetic variation that's there too. And I think getting to tools to manage that is something that it took a while to bring, kind of your average biologist around to. And now that we have, I think the future is incredibly exciting because, for me, to sort of answer this question about which is the right model, for that, I think it's the one that's fit for purpose for the question that needs answering, or, for the exploration that has to be made. And, I think one of the things that's been attractive about these cell models is that they allow you to grapple with the incredible genetic heterogeneity of ALS patients and to try to begin to understand whether or not there are convergent pathways that are really engaged by the many different variants that the community is finding. And it's hard to think about having a stable of 20 different mouse models that you would run your favorite therapeutic target through, but it becomes a little bit more manageable, although not inexpensive to Rita's point. But I think it's obtainable.
Yeah, so that's actually a good point too, with what Rita said about using multiple models, in the sense that, do you all encourage as well that you would use multiple patient types in terms of, I know that the Wanger paper originally was one of the ones that looked at SOD1, C9, several monogenetic forms, but then also sporadic. Is that sort of the encouraged way to look at this, so you can sort of parse out whether or not there's a personalized piece to it, or whether or not it's a potential generalized mechanism? Any Rita or Clive, or is that the way it should be done, or too tough?
I think in that, to answer that question, I would really sort of double down on what Kevin just said. It really depends on what you're looking at and what your hypothesis is and what the question is that you're trying to answer. And from there, you're gonna have to decide, can I just look at a monogenetic form of ALS, C9, SOD1, you name it, or do I want to look at something that, I want to see, is this happening in sporadic as well? Obviously, we're all super, excited, and we would love to know better and try and understand better what happens in sporadic ALS and what drives sporadic ALS, because that's the majority of the patients that we know. That's the majority of the patients that are out there. And while we're hoping to learn from the familial forms of ALS, we're also trying to then see whether we can translate some of these knowledge into the sporadic forms of ALS. But that's when it gets really complex and complicated. Because sporadic ALS is a whole array of ALS patients. We have not just slow and fast progressor, there are other differences as well, in addition to those two major terms that we usually use to differentiate between sporadic ALS patients, right? I mean, there's different pockets, different drawers, there's different types and flavors, and that's where it becomes really complicated. So it really all comes down to, what is it that you're trying to learn? What is it that you're trying to discover and to investigate, and then make a decision based on that. So I don't think there's an answer that sort of gives to everything. I think you really have to go by question, hypothesis by hypothesis, and then decide whether it's worth the effort to introduce additional lines. And then the question, of course, comes up, how many lines are sufficient lines? And obviously, if we deal with sporadic ALS, we cannot make isogenic control lines where we, then, say, okay, we remove the mutation, we correct the mutation, and we have an isogenic control that then can be used to really see whether that particular mutation was driving the phenotype that we're investigating. So it becomes more complicated, but I really think that this is something that we have to do on case by case, and really sit down and really think about this before we dive into this. because that saves you a lot of time and effort, and maybe we can use a 293 cell first in order to understand some of the questions that we have, and then come back and revisit this question using whatever iPSC line, whatever patient line we think is most relevant in order to validate what we found in a different cell model.
Yeah, I'll just follow up from readers if you like.
Sure.
I think the best papers that I read nowadays actually utilize, they'll do IPS and they'll try and get the phenotype, but then they go into hex cells to ask specific molecular questions and then come back to IPS. And that's a good way to do it because you can't afford to do IPS for everything, that's for sure.
Right.
So it comes down to the question. The interesting thing with genetic mutations is the amount of effort going on in therapeutic approaches now from Biogen, from others on ASOs, gene therapy approaches. We're starting to see some real movement in that field. You have to look at Huntington's, right, where we've known the mutation for 20, 30 years almost, from Jim Gazella. We still don't have a treatment, but now we've got better ways, better tools. So ASOs have come along, gene therapies come along. I kind of feel that the genetic versions of ALS have a really good hope that something will happen. The other 90%, as Rita says, is a real problem. What do we do with sporadic? Because we have no target. And I'll just mention Answer ALS, and that's something that you all know about. And we're making, this is with Jeff Rothstein and Merit Chikovic and myself, are kind of coordinating it, but it's a huge collaborative effort with just about everybody involved in ALS. Kevin helps as well. I mean, we're all piling in to try and understand how to do this. We're making 1,000 IPS lines from patients with ALS across the country. And you can go to the Answer ALS website and find out what it's about. But that's coming the question there is, if you take that 90%, is there a molecular target you can find by making IPS into motor neurons and doing omics? Maybe there's something that we're missing. And if you can find that target, then you can do the ASO approach with that subgroup of sporadic. And just what's interesting to me is how difficult this has been. I mean, Kevin and I have been doing this for how long, Kevin? I don't know. We had some of the first papers in this area. And it's been really difficult, to be honest, to find a phenotype in sporadic ALS lines. I think Kevin had one of the first papers with SOD1, and that's kind of simple, but not simple, but it's, the mutation, what to look for, right? And you see things. The sporadics have been a pain, to be honest. I mean, there's nothing like smoking gun coming out, and I've done a lot of differentiations for sporadic. You have to repeat it, and then it disappears, and you do it again, and it's there. So we need to really find out, and it's kind of telling me that, to a degree, sporadic ALS may be a lot of environment involved. I mean, that's where I'm coming down to. We did the same thing with Parkinson's, early onset Parkinson's disease, and we found a phenotype right away. It popped out. Didn't have to just 10 lines. And ALS has been more challenging. So I think you'll find maybe sporadic ALS is a complicated beast, and there's a lot of environment gene going on, maybe a little bit more environment. But that's just a feeling. We need to keep looking at the sporadic Answer ALS data set. We're making it open so anybody can access that. And I think there may be an answer in there that we haven't found because we're not clever enough to look at all that information. It's a ton of information, but anyhow, I'll stop.
The data browser is online, everyone. So you can go look up the data. I think so, right? You can look up a lot of the transcriptomic data, the proteomic data, all of the, yeah.
We've got some of it very early, though. There's only like 60 lines that you can look at right now. But we're very close to launching the second version of the web browser, which is in Azure. So it's going to give us a lot more power. That's coming, I think, around January 4th or 5th, we're going to launch a web browser that will have a lot more patients, up to 200, 300. And we're going to keep feeding into that with all the proteomics, ATAC-seq, and RNA-seq data that people can access. Right now, you can get limited sets of data and download them. But really, this is a very early kind of taster, if you like. It's an appetizer. The main course is taking a long time to get. We have to get it right. So one of the challenges is if we put crappy data up, people access it, they're going to complain like, oh, that was terrible. And that's a huge amount of back-end work to make it, all this stuff integrate. So that's been a bit of a challenge, but we're working hard.
So that actually, there's so many jumping off points there. But actually coming back to the idea of the sporadic and potentially some sort of environmental aspect, if we were to talk about the direct reprogrammed or the eye motor neurons, for example, are there places where those are being compared, like almost like a self-twin study where you're looking at the one twin who's had more environmental factors versus the IPS-derived motor neuron from the same person, and see if there's ways to see a phenotype from that, or is it Is it too difficult to still see a phenotype from like an eye motor neuron from a sporadic?
Because just, I think Victor needs to get in as well in a second, but I'll hand that to Kevin because he's made direct reprogrammed motor neurons. I think you're one of the only ones that have, Kevin. So, but that'd be a great experiment, David, to try and do that direct comparison. We haven't done it.
Yeah, I mean, I think these are really important points. I think it's important to remember that twin studies show that ALS is not a completely heritable condition, and that by definition, there are environmental components to the disease that, I think we only have a kind of modest recognition of, right? So, people have been worried about variant amino acids that can be found in certain environmental settings with cyanobacteria, injury, participation as a soldier in different military theaters. All of these things have been associated with ALS. Whether or not you're a smoker, whether or not you're a man or a woman has some modest, role to play. So we should be thinking about those things. The other challenge is the genetics itself, which I think recent studies begin to show us has a very long tail with respect to rare variant effects, and that I think nice work from our Canadian colleague Sally Farine really shows that there are probably variants that may only exist in just 1 or 2 people that have a meaningful effect on the risk of developing ALS in those people, right? And that effect is scattered across many genes. This means that it will take a long time and a lot of genetic studies to recognize that they're there, but that for a subset of the sporadic folks in Clive's collections and in these Answer ALS collections, there is a rare variant having an effect there. So we just don't know what it is, right? And this makes it hard to account for in our experiments, and we don't know which patients they're in. And then we know that there's also a risk conferred by common variants, but that as a community, we're still, driving the number of study participants that we need to push forward genome-wide association studies and measure those common genetic variants. So I think we need to continue to be supportive of these public health studies and of genetic studies, because they'll fuel the modeling and our thoughts about the disease. So, I think that is a really important topic. And then with respect to, kind of aging and things like that, I think my own preference is to kind of actually take advantage of the blank slate of iPS cells and to try to test hypotheses that are raised by these observations from our public health colleagues. That is to say, to take advantage of the fact that the motor neurons that we are making are young and that they have not been exposed to those environmental stimuli. That provides an opportunity to really ask whether or not any of those stimuli converge on the same pathways that are affected by genetic variants that cause the disease, right? And I think that there's still an enormously untapped potential for iPS cell-derived cell model studies to really take a disciplined approach at doing just that. I think, how does axon injury affect TDP-43 biology? What are the ways that neurons change their transcriptional profiles if you expose them to cyanobacterial components? Those are just kind of the, I think for me, really interesting things that could be engaged with and that are really made possible by the types of studies that Answer ALS is beginning to, provide a substrate for. So I'm quite excited about the future of that. As far as lineage reprogramming goes, probably our lab and Justin Achita's lab have probably done, more in this area than most. I think it's a very promising strategy. I think one of the things that can be hard about it is, the black box of aging is quite long. So, if you find something, there's not necessarily a straight line to understanding what it is from and drawing a conclusion about it. So you might be able to find the effect and begin to get there and kind of get a signature of what might be going on. But then you do have a long road to trying to deconstruct what got you there. And
what caused the signature?
I'm not saying it's not a full approach. I think it could be. But for the moment, anyway, I would say that we'd probably favor the former rather than the later.
Okay. Yeah,
I
it's
just-
actually interesting because I know that there have been some workshops lately talking about prevention of ALS, and in the sense of an iPS cell, it's, you know, there's almost an interesting piece there where you're given a naive cell that, in terms of any environmental impact, just the base, you know, the base genome to work with, and may lead to the potential for preventative therapies down the road, which is not something I think I see as credit given a lot, which I think, you know, obviously we're just talking about.
Well, I think that just to get back to what Rita said earlier on about postmortem. So we always get, when we submit a paper, they're saying, yes, but it's that early phenotype you see in these embryonic kind of, do you see it in postmortem tissue? And we're saying, no, it's the neuron's gone and dead.
It
happened.
Yeah.
This was a causative event, like Kevin was saying, that led to downstream things that happened over hundreds, over tens of years, because you set the neuron up for failure later on in life. And then I think, I'm thinking of it more like a Lipitor, right, with heart disease. You take Lipitor in your 40s, I'm already on it, to prevent something that's going to happen when you're 70. I think we're going to get there with ALS and Parkinson's, where you'll give a drug that we know stops something occurring in the cell. And it's probably something to do with the proteome or the lysosome, bad degradation of protein, because all neurological diseases are. If you can isolate that early, you know, the problem is those drug trials are very, very long, right? Because you're not going to know beyond my career whether it worked or not. But those are the kind of things young investigators should be looking at, because they have 20, 30 years to see if they were right.
Right.
Well,
and I think the genetic one. Oh, sorry, Vic, please.
No, I just want to step back and talk about the environment. I mean, the environmental factors clearly have a role in most, if not all, probably neurologic disorders that don't have a very clear genetic component to it. But for me, the problem is, how do you represent environmental in the lab, right? So you can, there's the obvious ones. I guess you've got formaldehyde, you can introduce lead and things like that. But speaking from my neighboring province, Brunswick, you know, there's pockets of really hot spots of ALS. This is a population of only 1 million, and it's a relatively, you know, modest sized province, but it's a large area. It's mostly rural. And the pockets tend to be around pulp and paper mills. But what is it about the pulp and paper mill? We really have very little idea. And the problem that it is, and we've got people looking at that, the problem also is, you know, people in the Maritimes, this part of Canada, you know, historically the families don't travel much. You know, they're poor travelers, and they like to stay home. You know, if they're in a fishing village, they're going to be fishing. And so, you know, you've got a real genetic component that you have to tap into as well. So you know, trying to find that factor to bring into the lab. And then the next question, and maybe, you know, Kevin can answer this better than I can, but, you know, is the, you know, if you can induce a mutation, where if you can induce a phenotype, with lead, is the same phenotype with formaldehyde, for example, is it all going to converge on the same pathway, or each one of these environmental toxins going to convert, you know, initiate their own pathway? And so I think, you know, that's, to me, is sort of the excitement, but it's also a bit of the maybe the problem and challenges going ahead.
Sure. Yeah. No one on any of these panels makes it seem easier. That we've got a lot to do with gene environment interactions, and obviously it you know, we've come so far in ALS, but it's just complex, right? So I think the reason why I say that too is we could really go off there and talk a lot about that aspect, and I'm happy to if you really want to. I also, coming back to the idea of you know, some of the skepticism in these cell types, and then where we are now, and then what someone who's listening, let's say it's a postdoc, a student, or even a PI who wants to work on iPS cells but is worried about, you know, batch to batch variability. Where are we at in terms of harmonization of protocols? What's the best protocol to use? Should they be collaborating rather than doing it on their own? And then if they were to do something on their own and say, look, I'm going to set it up in my lab, either for a particular experiment or something I want to do, you know, moving forward, what are the key things that they should be aware of? Because you all have so much experience in this, in terms of doing these things right. What pitfalls could be avoided? As an awful, again, loaded question, but maybe just collaborate with people who do it already? Is that like, 'cause seriously, I think in some cases that might be a valid answer, but
you know, I think first of all, these things are getting easier to pick up. And, you know, I think companies making medias and, you know, more available cell lines like from Answer ALS and other sources are making the barriers to entry lower. That's good. You know, but obviously, you know, it's still complex cell culture like any other complex cell culture. So I think having a path to learning it and getting exposure to it from an expert can be enormously helpful. And then I kind of like circle back around to, you know, to this issue of what are you trying to achieve, and, you know, how long do you want to be at it, right? As being pretty important with respect to guiding that kind of strategic decision, right? And, you know, I think it also depends on the type of biology that you're working on, right? So I think maybe some crisper, clearer, you know, kind of, you know, kind of experience or kind of cases might be helpful, right? So, you know, for certain types of pathway biology, you know, maybe around regulation of the proteasome in the broadest sense or things like that, you know, much of that machinery is pretty well conserved in all human cell types. So if you wanted to study something about that or begin to get a sense of it, you know, you might be able to do a first pass experiment in other models. You know, it wouldn't completely protect you from the possibility that there's an alteration in that cell biology that was unknown in motor neurons, but it would be a logical place to start. You know, in contrast, you're not going to get very far in, you know, trying to identify pathways that regulate ion channels in a 293 cell. You know, I mean, I think you've got to be kind of aware of that. But you might be able to study that in a rodent neuron and be able to make some progress. You know, I think for us, you know, really it was TDP-43 itself that kind of got us really, you know, I would say stuck in on these types of models in the longer term. And, you know, just the realization that that is an RNA binding protein, you know, has really major roles in regulating RNAs, and that in general, RNA regulation is a surprisingly poorly conserved process across species, and that, you know, splicing patterns and factors that regulate transcript biology are quite divergent. And so it just sort of kind of followed for us that if you're going to really understand what TDP-43 did in, you know, human neurons and how that was changed by pathology, that you need to do that in a motor neuron context. And, you know, probably even a bad motor neuron was going to be better than a mouse motor neuron, given the lack of conservation of RNA metabolism in that setting. So like, that's a very like granular example of like why we felt we had to do it in that context. And, you know, and that's the story of Stathman 2 and why that kind of popped out. And, you know, once you know about it from a model, it's really easy then to go look at it in patients and see, wow, that's a big change. And we weren't really quite powered to detect it in a genome-wide manner in a screen. But once you kind of knew about it and you had a hypothesis to test, it just jumps right out at you, right? So I hopefully those are some like practical, granular, you know, examples. You know, you can never account for the complete unknown, but if you have a very clear hypothesis about, you know, something that you know drives you to a particular type of human cell, then, you know, that may be the thing that triggers you to take the big step.
So I'm gathering though that it's largely like thinking, and from what Rita said earlier too, thinking about your experiments very carefully and deciding whether this is the avenue that's going to help you prove what you're looking for, and as opposed to something that's cheaper, easier, and probably get you to the same answer. And so these are, you know, there's a lot to be, and as we move further with the sporadic side of things, these are going to become, you know, really interesting if we start to solve a lot of the monogenetic, more penetrant forms with therapies. But beyond that, it's really thinking carefully before diving in. But so then for reviewers, should that be something that they should know too? Like that you just don't automatically go to where's your proof in a differentiated human cell type, or is that probably pretty well known, you know, felt across the field, do you think?
What do you guys think?
Or do you think there's still a need to do it to get into a high impact journal?
Yeah, I mean, I don't want to talk down on any reviewers just because of bad experiences, but it's certainly, I think we all came across, you know, some of those comments, obviously, either through grant applications or manuscript submissions that we had, where then a reviewer says, well, why didn't you just repeat this experiment in iPSCs or in additional iPSC lines, because you only use 2, but we would like to see it in 10. And then you're like, well, that's another 5 years of work if that's what you want me to do. And is that really going to add more to the data and the information that I'm providing you as is? So these are very, you know, difficult decisions to make. And I think, I mean, I want to see that, or hope that most of the reviewers that get to review these kinds of manuscripts or grant applications have some experience with iPSCs and they realize what, you know, what the values are and what the benefits are, but also what the difficulties are with these models, and keep that in mind. I mean, I certainly do when I review grants or papers, and I don't lightly just say, hey, I want you to add a few more lines, or I want you to actually show this in human cells. So I really think about what Kevin sort of hinted at. I think about, is it really necessary to show this in a human neuron, or is the cell or the model that the person has been proposing going to be a good enough model to do exactly what they're trying to do? Or is a human cell, a human neuron, a human glia, is that necessary in order to really get to the answer that you're trying to get to? And so that requires a little bit more, you know, sort of really your self-check when you're reading this, to see, well, do I really think it is important to have this? And when I feel that there is an importance, then I will put that down, and I suggest it might be good to potentially add some human data to this. And coming back to what you asked before, for people that are new to the field, is it worth collaborating? And I say yes and no. I mean, if you really just want to answer one question, then it might certainly be worthwhile maybe reaching out to somebody first to initially see, hey, is there any value in us using these models by just running, you know, it's always easy to say run a couple of experiments, because that's never just a couple of experiments if you do human cells. And speaking for myself, I certainly have been bombarded by, you know, people just asking, oh, can you just do a couple of experiments and see how this works in a human cell? It's not as trivial, but it's doable, and we certainly encourage people, and I do it myself for other models that I don't have running in the lab. I reach out to people that have other models and say, hey, would you be interested in working with us on this? Because we would like to see how this looks in a different model so that we can compare it to our models. So I don't think people should sort of, you know, be afraid of doing the neuronal stuff or the human neurons and the human glia, but maybe go baby steps and maybe see whether there is somebody who wants to collaborate with you. Maybe there's somebody who allows you to come to the lab and learn it in your lab, so that you have somebody that sort of holds your hands in the beginning, and you're gonna have to, you know, you're gonna get this taught by somebody. I've done this. I've sent people, when we started doing different glia differentiations, I sent my folks to a lab that has already worked on those glia differentiations, so that they can learn from that lab the little itsy bitsy details that you don't see in a manuscript that's published. But maybe really, before you jump into this, because it is a big effort. It takes space for tissue culture. It takes, you know, a really, you know, a very talented, you know, technician, or even, I mean, it's hard for students and postdocs. This is a lot of effort that they put into this. That takes a lot of effort out of their work, and you know, they have a restricted time that they want to get their PhD thesis done, or they want to finish their postdoctoral study in your lab. So you're going to have to be also sort of aware of the restrictions, the time restrictions that people bring with their different career paths that they're going for. And then, you know, maybe it is worthwhile just saying, hey, can we just look and see whether this gives us truly a better answer than what we're doing currently, and then make a decision whether you want to establish this in your own lab for young investigators. Is this something that I want to integrate in my lab and dive into and really set it up as one of the models that we're studying? It's not an easy decision to make, so it's.
I think on the
I think it's great. I think what we can say here is that the people who are very pro these models says tread carefully about jumping into these models unless you need these models. And so I think that's great.
Can I add something from the Canadian perspective?
Sure. Of course, yeah.
Echo, I mean, you know, they're a lot of fun. That's one of the reasons why I do them. That, you know, I think they're very valuable, but they can be quite rewarding, and they're fun. And certainly, it's a great recruitment tool for graduate students and postdocs if you're doing that type of work in your lab. But I completely would echo what was said, is that I think you would start off with collaboration, and it's expensive. I mean, you know, when you're working with a single CIHR grant, it's a big chunk of that grant. And if you have an NSERC grant, you're not doing it. It's just as simple as that. So, so I think you have to think about, you know, cost benefit. But, you know, I wouldn't shy away from doing it. But again, I think the common theme by everybody is, what's your question? And is this the best technique for the question? If it's the only way you can answer it, it's the only way you can answer it. And my final statement before Clive jumps in is I'll age myself, but back in, you know, in the 90s when I was doing a lot of chick work, you know, you would show this stuff in the chick, and then they'd turn around and say, well, you need to now show it in the mouse. The reviewers say, well, you need to, and then you had Tom Jessel, Sam Pfaff would champion that and sort of set the stage, and that's what they did a lot. But it put, you know, other labs maybe at a bit of a disadvantage, even though I would still argue that chick's a valuable model for developmental issues. And so I think now we are reaching a level where if you show it in mouse cells, and you don't show at least something in the human cell, you might be at a disadvantage with reviewers, and I can speak from experience.
So I'll just say a few things about what I've heard, and I again agree with everything. It's been a great discussion. By the way, this is a great panel, guys. I'm learning. You're always learning in science, right? But to me, the reviewing is a pain. I'm sure I already review a lot. I see two different buckets, right? So there's discovery IPS work, where you're trying to say, oh, there's a new marker like Snapman for ALS. You have to be a little stricter there about numbers. And, you know, we're getting to the point of like 10, if it's sporadic, you have no mutation. So 10 is becoming a magic number, 10 and above, to at least start saying there might be a difference if the effect size is pretty big. So that's expensive, it's a lot of work. Discovery is a lot of work, and even then it's probably not enough. But if you're getting into the specific questions, particularly with the mutations, I like the papers now that have 3 lines that are isogenically corrected. And then I like the papers that challenge those lines with something, so you have a baseline. So now you've taken 3 lines, you've got your isogenically corrected ones, and now you challenge, say it's SOD correction, and now you challenge them with different stresses. So you have a baseline. So one line, Kevin and I know this, you know, one IPS line might make 30% motor neurons, and the other one only makes 10%, right? So if you just average those 2 lines, you're going to have an enormous amount of variation, because if you just took the total number of motor neurons over time. But if you start with one line and normalize to that 30% and then stress it, you have a much more powerful statistical tool, and you can take the isogenic and say, was it due to that specific mutation? So the easier experiments to get into really are the ones that involve C9 or SOD1 or TDP mutations. And those ones you can really, I think, design nicely and get away with smaller numbers of lines. The complex ones are where you're in sporadic area, because you got to convince people first that you even have a phenotype. And I'm not seeing many papers with sporadic that give you a nice phenotype. At least in C9, you have the dipeptide repeats, you have inclusions that you can really say, you know, we reverse that in anything that you do, because you need a phenotype, right, of some sort to kind of work back from. So that would be my sort of 20 cents. And I think collaboration is great. If you see a paper with the lines, they should be able to help you get those lines, so save you making them. Last thing, a warning, and again, we all know this, is not all lines are equal. If you get a line from a lab that's at passage 40, it could already have trisomy 12, it could already have a whole lot of stuff. And your grad student spends a year, and then, oh, I karyotyped it at the end of the experiment. I've had that before, believe me. Many, many tears, because it wasn't a normal line. And these lines do pick up aberrations, and depending on how they're made, could be up to 25% of the differentiations you do have an aberration of some sort. So That's the tough part of this job, is trying to control for that. And it's expensive to karyotype every so many, you know, passages, but those are the sort of things that there's no easy answer. You just have to be very careful and cautious. And when you do.
Oh, I think we lost the sound, Clive.
You finally get a clone that's beautiful after your CRISPR editing. That's when the stress that you put it under to get that clone sort of brings out this abnormality, or karyotypic abnormality, possibly. So there's kind of a slight correlation there. So those are kind of the warning signs, but definitely get into it. And Rita said, if you have a specific question that can add to your paper, jump in.
Yeah. Well, this is great. So I'm actually going to take one of the next things I thought about saying, and the one after that kind of merge the two, and the idea of like what's next. So most of the work, it seems like, has been done in terms of this type of cells, turning them into neurons, looking at a purified neuronal culture, for example. All of you have work on other cell types, whether it's muscle, whether it's astrocytes, microglia is now something that can be derived, other neuronal subtypes. So even Rita mentioned before about not just fast and slow progressor, but what about cognitive spectrum and the need for cortical neurons? And then the question is, what's next in terms of, do we need to be incorporating those other cell types and glial cells, for example, into the models? And then what about models like organoids, assembloids, body on a chip? Should we be letting other fields take that because ALS doesn't have enough money until it's optimized? Another loaded question, but I'm going to wind you guys up and let you go on some of that as to what's next for these models. Anyone want to start?
Come on, Clive, you're doing the chip stuff, so why don't you get started?
I'll go first, and then I'll hand it off, because, you know, and I have to say some of Victor's work is absolutely beautiful of getting this sort of maturation. So when you do motor neurons, you know, they're not floating in your dish, right? They're connecting to a muscle. And I think we always forget that. And Kevin and I have been doing this for years, but, you know, and so it always worries me that we've got this motor neuron in isolation. And when it extends to the muscle, it upregulates peripherin, it makes an axon that's peripheral, which is part of the whole process. So I think the kind of work Victor has done, we should be thinking more of incorporating those, even putting muscle juice in or something to activate those motor neurons. And we're moving one step. So we're trying to, the other big missing part, you mentioned astrocytes, microglia, is the vascular system. There's no neuron that's more than 40 microns from a capillary. And so we've been focusing on that for the last few years, and with this organ-on-chip technology, where you can seed neurons under the top layer of a microfluidic device. We use PDMS devices. They have little holes punched in them that are 7 microns big. And then on the bottom of that device, you have another tube on the bottom and a tube on the top. The bottom of that device, you seed endothelial cells from the same IPS line that you make, brain endothelial cells, BMECs. And they actually stick around that PDMS material to make a mini blood vessel. So now you have this blood-brain barrier. We accidentally made it, and we thought, oh, that's pretty cool. And so we thought, oh, we actually made a blood-brain barrier. We flow blood in the end through that bottom channel. And now you have a much more physiological system. So you can throw microglia, astrocytes, neurons into the top. The astrocytes put little end feet onto the blood vessels through those 7-micron holes as well. It's not perfect. It's not recapitulating the beauty of vasculature of the brain, but I think it's getting you one step closer to heaven, you know, which is the perfect model, right? And finally, but I'm still back to what I said when I started my first comment, validation. I don't care how beautiful the model is, it may not be accurate, and it's not going to validate until I put a drug in that blood and vessel that activates the neurons, and then I give it to the ALS patient, and it has a similar effect. Then you've got me. Until then, it's just another model. It's really fancy. It's a chip. Still a model until we validate, because we're not, you know, unless we get, you know, organoids are great, but they don't have vascular system yet. So I feel like they're a little limited. Unless we get that, and we don't have muscle in our system, Victor. I'd love to get a muscle in the chip as well. And now you're talking about what Kevin, you know, you could attach the gut to the brain. You can sort of do this stuff, but boy, that gets complicated. And you're never going to reproduce the exact physiology of the human. So my question to you guys is, how much do we need? How far do we have to go to have a model that validates a drug in an ALS patient can help them. And I think that comes down to having a good phenotype as well, something in those systems that could predict ALS very well. And we don't have those yet. We're still working.
Would the presence of other cell types possibly push sporadic towards a phenotype?
We live in hope.
Yeah, definitely. I mean, I think that Victor's work, many fields have pursued this goal through xenotransplantation studies. And so, I think in our field, Victor has really emerged as the leader in that area, just full stop, and, I always kind of wondered, just for sure exactly what we were making until we teamed up with Victor and started shooting these cells into rodents and saw just how amazing their ability to project down through the peripheral nervous system and reinnervate muscle and keep it strong, and so, I can't help but wonder whether or not we should be making, humanized animal models where, we're accepting that there are some things that we need to sacrifice potentially with respect to, immunocompromising those animals to get it to work, and that could raise some questions about your ability to study neuroinflammation in that context. But, the ability to transplant a human cell and to have it, kind of set up in even the peripheral nervous system and some of the approaches that Victor's really pioneered, in my view, would allow you to make, a motor neuron that's a couple years old. And it'd be a heck of a lot cheaper to have it running around in the cage rather than having to feed it every day. Mice aren't free, don't get me wrong, but, you could imagine all sorts of interesting experiments in that context where you have ALS motor neurons on one side, and then, on the contralateral side, you have the control. So everything about the experiment is, internally controlled. But there's a lot of cool, places that could go. That kind of takes you all the way to the end, right? That's the most complicated model that you could think of, right? Where all sorts of biology are present. And then you could try to take that apart through more complicated cell culture systems like the one that Clive has talked about. So I think that's a really exciting area. I think we should definitely hear more from Victor about that.
Well, I'll take the compliments from you two people any day. So thanks for that. I'm a neuromuscular person, so that's what drove those experiments. And Kevin and I have talked about what sort of the experiments he described earlier, because I agree. I mean, once those motor neurons have made functional contacts, they, at least in our hands, I mean, we've gone 9 months and they stay pretty healthy, and they form very normal looking neuromuscular junctions from all, anatomically and electrophysiologically. I think the stumbling block we've always had a little bit from jumping into that is just the immunosuppressant. So putting an animal on immunosuppressant for that long, in our hands anyway, it's problematic. They don't tolerate it very well. So you might get 2 months out of it. So there may be other ways to go about it. Choosing different type of mice. But it's definitely something you retweak my imagination, Kevin. So I may revisit that. I mean, the other thing that Clive sort of mentioned. I mean, none of these are, when you differentiate. I say, well, I differentiate them in the motor neurons. Well, they're not. You don't get 100. At least my hands, and I'm hearing from Clive, and I'm sure everybody else, right? You get a percentage, right? 30%, you're kind of happy. So you get a 70%. What's the other 70%? And we always call them interneurons, because final interneurons, because it sort of would make sense, right? And we've sort of, we've got SIM1 positive, so we know they are at least a subpopulation of them are. But what I think, when you're putting together these systems, they're already a complex system in themselves, because we showed a couple of years ago, really to our surprise, that even in these transplants, after time, the transplant itself, sitting in the peripheral nerve, forms its own central pattern generator. And so you can give it a blast like you would a isolated spinal cord prep, embryonic spinal cord prep. You give it a blast and it starts walking, and the exact same thing happens. So, and it can be blocked with glutamate antagonists. So you already have a complex system, even though we don't think about it. There's clearly interneurons there. We know that the interneurons and the motor neurons interaction is not normal. in ALS. So, there's something there that's going on. And the muscle side, I'm a neuromuscular physiologist. I show my bias. I do think the co-cultures have something to address. But in our hands, if you ever looked at, read my papers, you'll notice that the muscle we use really comes from a very specific subtype of muscle in chicken embryos, right? So, and so even the different types of muscle fibers in a chicken embryo, whether it's grown with Kevin's human iPS cells that we collaborated with, or with most iPS cells, derived motor neurons, they behave very differently just with the type of muscle that they're sitting on. And so we've been mostly successful one subtype, and we've had very, very, very limited success, although we've tried many times. And if somebody out there can help me with this, with ES cells or iPS cell derived muscle fibers, I mean, they kind of make neuromuscular junctions, but in our hands, they're just not super satisfying. So anyway, I just rambled there a little bit, but hopefully I addressed some of the things. But certainly, the in vivo transplants could solve a lot of this aging problem.
But I think that we should take that as a hint that something is missing from those muscle protocols, Victor. I mean, I think, when you can do it in one context, but you can't do it in another, it means, to me, I think it's like, at some point you do it enough times and you start, beginning to develop some confidence. And actually, I think that's one of the hardest things maybe for the trainees out there to kind of start gathering intuition around, is like, when is something not working telling you something about the biology, right? And, it's very natural, for everyone in my lab who comes to want to try to use these, what are these C2, C12 cell lines that have been out there, and yeah, they kind of make something that's like muscle, but I always, point them to Victor's work, and I'm like, why don't you just go crack some chick eggs and get some real muscle, and because you'll have a much more satisfying, kind of experience. And that blew me away, seeing the muscle contraction that, iPS cell derived motor neurons could drive, and that, chick just blew my mind. And I think that to some extent, it's become kind of an engineering problem, because the preparation just rips itself off the dish, because the the contractions become so violent. So it's not a little bit of this kind of wimpy barely contracting stuff when it's really happening. And so you kind of it when you've got it. And I think trying to figure out that vascularization, a great goal, could really help with stability of culture in the long term. I think purifying just to purify is good for molecular analyses, but isn't good for long-term culture of the cells, where they're used to having interneuron inputs, other types of cells around. Clearly you need astrocytes to promote synaptic maturity in different contexts. There's a bunch of components to think about here. And again, a bit of it's trying to not make it so complicated that you can't ask your question, but recognize that there may still be opportunities for biological questions to be answered if you can overcome a technological hurdle that someone else hasn't.
And quickly on the muscle, mastocytosis muscle in my lab when I was in Wisconsin was trying to make muscle from iPS. It got something close, but it wasn't epithelial muscle. And I think you've got two imperfect systems. You've got an imperfect motor neuron and an imperfect muscle, and two imperfects make a really crappy model. So I think one has to be anchored in a real life. So if you can get human muscle fibers from a biopsy, that would probably be your best bet to then attach your developing muscle to. And the secret there is development, right? If you look at many papers, they tout that, I cured ALS by connecting something to something. Well, they've done it in a developing system. When you get to the patient, if you ever want to replace a motor neuron, our challenge clinically is this is now a motor neuron that's 6 foot away. 6 foot. I'm tall, but I'm not that tall. 3 foot away from its original target. And I think that's always haunted me. I've always been excited about trying to replace a motor neuron. But putting it back in the cord is always a daunting challenge in an adult. Could you short circuit it, Victor, by putting it in other areas? I mean, but we should keep trying. I don't think we should give up. I think this is a goal as a field, and you've got to reach high and take the appropriate steps. And I think we will get to the point where maybe we can replace an ALS a motor neuron and have it reconnect to the muscle. I mean, how good would that be?
Well, that's the the Don Cleveland thing now too, with the astrocytes being turned to neurons, and then I think was Linda Greensmith had that one where they put it locally or something with optogenetics.
Yeah,
Marty Bryson.
So a neuron is sort of sacrilege for people like Mike Sofferny and myself, because the astrocytes are more important than the neurons, guys.
So we have like 10 minutes, but I want to ask one quick question. But like, so basically, in summary, we've got it all figured out. It's easy. That's what I'm hearing from all of you. No, we're in the middle of a long optimization process with a lot of value to the right experiments. Is sort of what I'm hearing. With a lot still left to learn. And just tell me I'm wrong if that's wrong. But thinking about therapeutics now, very, very quickly, because I know it's a big topic and we're not going to hit. But so I'm hearing that obviously we still haven't quite got a point where you've got sporadic that are getting a phenotype enough, or at least biomarkers enough in the lab that are going to allow us to say, do a clinical trial replica using these cells per se. But then you have something like Teralis is working, I think, using these to try to advance autophagy and the sodium potassium channel aspects. Is this, in terms of drug discovery, are these cells ready for the prime time? Or do you think that this is the thing that people should be validating in this before going to humans, or even using it as a primary model to jump to humans for even novel things that haven't already been FDA or EMA approved? Or if we still don't know? Clive, I think you're muted.
I would jump in here and say, just the path to translation is really distinct from discovery science. And again, here it's highly contextual. So, I'll just provide one frame, right? So I think that getting to early oligonucleotides, that had clinical promise, and going from idea to hit oligonucleotide therapies to lead oligonucleotide therapies, for sure went a lot faster for Stathman-2 and went a lot faster for NR72 because the ability to have human cell models than, for instance, they did for SMA. Because, there you had to build this mouse that could do the human biology. And, there where you're developing these nucleotide selective agents where you have to have the human gene, it's not that people aren't building Stathman-2 humanized mouse models. They are, but that takes time and it sets you back years and years. And so we'd still be waiting to even get started instead of many groups having those oligonucleotides already in hand. So, it's probably saving you a couple of years by being able to begin the work. Now, it's still nice to have an animal model because there are questions that are harder to answer. Like for instance, does my compound achieve the required concentrations in the brain at a concentration that is also not toxic, which would allow me to assess whether or not there is a reasonable therapeutic index for my compound. And while I think it's exciting to think about putting human cells into animals, a la what I think Victor should definitely be working on, because then you could do your in vivo pharmacology studies in that context without having to make a transgenic animal. You could just take your human cell model, put it in a mouse, and see whether or not you get target engagement in the mouse at a desirable concentration while looking at toxicology. That's an exciting kind of future, but that's still kind of the future. So I think there's a little bit of back and forth here, but I think for screening, for discovery of molecules, for these early steps of translation, there's no question that it's ready for prime time. And you can see that it is because, basically all the companies are starting to use it because, they recognize that it's probably faster and cheaper than building a bunch of animal models. But, when you need to start asking questions about how your drug behaves in the body, these are still cells in a dish, and that's going to hold you back.
Yeah, just quickly, because I know we're running out of time. I'll give two examples where it actually is being put into practice, other than Kevin's, and he can talk about his, but there's two others. One is from Kano's group, Nature Medicine paper, where they actually did a heroic number of right in Japan. Ropinirole came out of that. That now, as far as I know, I talked to him recently, they're going to test it, and maybe Kevin has thoughts on this, but it's an example where they use screening and they did it, right? And Kevin did it with GSK company. He can talk about that briefly. And the third one is Acheta, and the Acheta group did something similar and came out with a small molecule and is now moving to the clinic. So I think the answer is yes. People are using it, and if any of these hits that they found in the IPS models convert into an effect in a patient that's better than what we do now, which is pretty lousy in ALS, then we'll be off to the races, guys. And you can come back and you can do your mechanistic studies, and you can add to it. And this is going to be revolutionary. I mean, I'm in this field because I think it will. When I was next to Jamie Thompson when he simultaneously discovered with Yamanaka how to do this. I have to have a pitch for Jamie here, and that Yamanaka would never have done it if he hadn't had all the embryonic stem cell experience from Jamie for 10 years. So they're both fantastic groups. But as soon as I saw that IPS technology, and it really is based on the work kind of that Kevin did and Wilmot, this reprogramming, guys, is so exciting. I mean, come on. You talk to people in the street, do you know what IPS cells are? Nobody knows what these things are. They don't realize it's been a revolution. And yet the whole field, the whole pharma industry, everybody's turning to it because now we have human models in the petri dish, right? We have a brain biopsy. We've never had that until 10 years ago in Yamanaka, and Jamie Thompson did that work. So, and Kevin, a lot of work that Harvard did came in as well, Melton. There was a lot of, it wasn't just Shinya, it was, as Kevin knows, the history is very complicated and interwove with a lot of Janish's work, and of course Gurdon, who got the Nobel Prize as well. So this is coming from very, very strong roots. And now we're reaping the rewards. And I totally get Kevin's kind of like this, like gene therapy, it went down. I think we're in this phase now. From these papers, you're going to start seeing some very exciting discoveries, and we're done. I mean, IPS are here. They've revolutionized medicine, and we're going to make them more and more like a human. And we might, in 20 years, have a human model really working well, like Victor wants, that is all in vitro. We don't have to kill any animals to do it. So I think that's the other impact. It's going to reduce the amount of animals we need in experimentation eventually, once the FDA is happy. So that's my ending.
I love how you brought it back to excitement, from complexity to super excitement. Does anyone want to add? We have a couple minutes. I have one last question. I think maybe a silly question, but I think it's a question that, okay, I'm going to ask the question because no one's jumping in. So with regard to then, this is not taking it from human cells in the lab, but thinking about the ability to take patient-derived cells. Now thinking about, there's a series of different therapies that are in the clinical development stage. Clive, you have one of them, which is looking at using stem cells as a vector to, in fact, you had surmised this a long time ago as a potential vector to deliver supportive activity to the motor neurons. Is there a possibility in the future where someone gets a personalized medicine form of that, where they get an IPS-derived astrocyte or cell type that's their cell type that then is used to go back to them as a supportive mechanism for the motor neurons? Or do you think that those cells will become obsolete eventually and we'll start to find other ways, AAV or whatever, to provide the same factors? Like what's sort of the future on that? And I asked that with 2 minutes left, which is an awful thing to say, but.
The key for us is whether the cells will reject or not. The blood-brain barrier is a wonderful thing for transplants because, like fetal transplantation, they took the immunosuppression away. These grafts lasted for 20 years, and they still don't. So I don't think we need autologous, to be frank. I think an ALS study we'll publish soon shows that you get cells surviving up to 3 years in the spinal cord.
Wow.
At least, yeah. So I think that's kind of the way to go. I think autologous is not necessary for these transplants, and that's in my view.
Cool.
And also, then you've got the risk finally of the patient has the disease, so the cells you put in are carrying whatever caused the ALS.
Great point. Great point. You'd have to find a way to replenish that anyway at some point once they become less supportive. Okay, does anyone, we have 1 minute, anyone want to add a final stamp on this? Or I mean, these things go so fast for me. I really appreciate your time on all of this. I'm so appreciative of all what you've provided. I think the audience obviously will be as well. I think obviously there's so many things that you could talk about but this could go for hours and hours, and I'm sure you're glad it hasn't. But nonetheless, thank you so much to all of you.
Kevin would say, I think Kevin would say something.
Yes, someone was going to say something. No?
I think Kevin would like to say something. It seems like.
Oh yes, please.
Oh, well, I was just going to say, David, thank you. I mean, I think this has been a great venue with, people's, whose opinion I truly value. And, I just feel like I got a lot of it. So I hope that's true for everybody else. Thanks for doing such a great job of being prepared and, with super questions that were right on point. I just really appreciate being included.
Yeah, I would echo that.
That's
David.
awesome.
Thanks for
You
thinking
guys
about.
made my day. Thank you so much. I'm a big nerd, so I love all of this, and thank you. And thank you to Biogen for sponsoring this. And we look forward to having everyone out there come back for the 4th in our series, which will be around drug treatment targets. So in ALS, with another stellar panel of 4. So thank you all. Have a wonderful rest of your day, and hope to cross paths with you in person someday in the future.
Sounds good. Thank you.
Take care. Thank you.
Bye, everybody.
Everyone.
Bye. Thank you.